Right? Steve is so happy. Anyway, welcome back, everybody, to another Journal Club brought to you by the Life Extension Advocacy Foundation. I'm Oliver Medvedik, broadcasting, I guess, somewhat live from Cooper Union here in the heart of New York City at the Cambar Biomedical Research Laboratory, of which I'm the director. Joining us are Steve, Victor, and Sven. And we will be discussing a paper that's a little atypical for us because it is not an aging paper, a longevity paper per se. It is a techniques paper that is really, well, it's not really a techniques paper. It's a paper employing pack biosequencing to look at potential of mutagenesis using the CRISPR-Cas9 system. So the reason we're doing it is because there's been a lot of interest around this paper. Obviously, CRISPR-Cas9 is a leading tool for targeting genes since its development, from the Doudna lab in 2013 and many other labs, and it's taken the world by storm. But there's been some issues in the past where people have raised I guess, worries about off-site targeting of this system. That's been dispelled, but now we have a new paper out that seems to, they do a pretty thorough job looking at areas of CRISPR targeting that are a little bit more local, and they raise some, serious issues, and we're going to see whether or not these issues really are that serious and if there's any workarounds. But bottom line is it's a pretty solid paper. But before we go into the paper, I think maybe we should just kind of backtrack a little bit and go over the CRISPR-Cas9 system for people who don't know what the heck I'm talking about. So this is not going to be a thorough overview of CRISPR. I'm just going to mention what it does. CRISPR-Cas9 is basically it's a ribonucleoprotein complex that was identified in bacteria. It's found in many bacterial and archaeal species, and it's basically a bacterial acquired immune system, and it's been essentially developed primarily since around 2013, so it's a fairly recent system, to programmably cut locations within genomes, and genomes of pretty much the entire kingdom of life. So any kind of organism that's out there, there's a paper out there, highlighting how CRISPR-Cas9 can be used to cut it. And there's many different variants of CRISPR-Cas9. The paper uses one, but you've got many variants from different species. So what it basically does is it cuts DNA. So it's an endonuclease that the native form basically cuts in both strands, and there's a little RNA component called the gRNA, the guide RNA, that guides the protein component, the Cas9, or the CRISPR-associated protein, to the locus, and that protein component does the cutting. So basically, you've got the RNA part that does the guiding, that takes it where it needs to go, and then you've got the protein part that does the cutting. And there's been many variations of this system built that, you'll find versions that have been synthesized, synthetic versions that can, modify DNA, not just cut it. But we're primarily concerned here with the cutting function of Cas9, and particularly the double-stranded cutting functionality. And the reason why we're interested in this double-stranded cutting functionality is because that's really the heart of gene therapy. If you want to modify genes, the most efficient way to do so is you need to cut the DNA at the location that you're interested in, and at that point, one of two things can happen. Either the DNA is repaired using a mechanism called non-homology end joining, which is error-prone, and that's particularly what they're looking at here at this paper, or you have homology-mediated repair take over, and this requires a template DNA, which then can basically be copied into the break site, and that's not what they're looking at in this paper. But suffice it to say, whichever direction you want to go, you first need to cut the DNA, right? So the issue with this CRISPR-Cas9 system is, okay, you cut DNA, the guide RNA, it's a little itty-bitty piece of nucleic acid that the whole length of which is, I think, roughly around 70 or 80 bases. 20 bases of which are really the key that homes to that specific location in the DNA. There's a bunch of, sites that, there's a lot of other, nitty gritty details we won't go into, such as protospace or adjacent motif and all that other jazz. But basically you have this 20 base piece of RNA that forms a heteroduplex and guides your essentially your protein scissors to that location. The issues that were raised early is, can this system go accidentally to other locations? Short answer is yes, and there are ways around that. You could make the guide RNA more specific. There's modified versions of the Cas protein that cause it to bind with less affinity to slightly off sequences that, so the sequence has to be very, very precise for it to bind. You've got, Cas9 variants that have been developed that use 2 guide RNAs to cut into slightly staggered locations to give it even more efficiency. So there's been a paper that was published a while ago which suggested there might be problems with off-target cutting, but I think it was an artifact of the system they were using, and it turns out that that's not so much of an issue with problems with the Cas9 CRISPR system cutting somewhere else, like way out in other chromosomes and in other regions of the genome. So that doesn't seem to be too much of an issue. So this is kind of cutting like really kind of far off. And in a nutshell, what makes this CRISPR-Cas9 system so awesome for people, because we have other programmable endonucleases. You've probably heard of TALENs, you've heard of zinc finger endonucleases. Well, the part that does the homing is the guide RNA, and RNA and nucleic acids in general are really, really, really cheap and easy to synthesize these days. So if you want to program your endonuclease to go someplace else, you just synthesize another piece of nucleic acid. And, the hard part is really the cloning at this point. But the actual, assemblage of the Cas9 CRISPR system is pretty easy. So that's led to a lot of labs now adopting this technology to do all of their genetic modifications for that reason. The paper that is just published now, though, looks at, it's a pretty solid paper, and there's nothing seemingly wrong with the data, but they look at something a little bit differently. So they're not looking at mistakes that might happen way out somewhere in the chromosomes, but they're actually looking at things that may go awry kind of within the location of the cutting itself. Now, when CRISPR, I mentioned briefly, and the Cas9 component cut, if you're not doing any type of homology-mediated repair, depending on the cell type that you use, you've got this NHEJ pathway, which tries to repair. It basically ligates the sequences together, but you have a lot of error. You have what's known as indels, insertions, deletions. And these have been typically thought to be pretty small, on the order of around, tens of bases. So you have basically little bits of DNA missing. And this is actually a positive, for people who want to disrupt genes, because if you want to knock out a gene, you put it in your CRISPR-Cas9 somewhere 5' of the gene, somewhere upstream, you cut it, and you get these insertion deletions, and a lot of them are going to lead to frame shift mutations, and you basically destroy your gene. But without any far off effects, right? So you don't have any, you hopefully don't have big chunks of DNA missing way upstream and way downstream. But what this paper has done is they've actually used PacBio sequencing, which is basically a sequencing technology that's been around for a number of years. And it utilizes essentially, my understanding, a very, very tiny array of reaction chambers that uses captive polymerases. And you basically use DNA fragments of your genome that have ends that are ligated. So now you have this little circular piece of DNA, and you can sequence fairly long pieces. I think the PacBio system does about 2 to 4 kb, something like that. So not short reads, but fairly long reads, kind of like Sanger dideoxy sequencing. So they used, in the paper, they used this technology to basically sequence the heck out of, a couple of bases surrounding where they did these cuts. And lo and behold, they find, a pretty significant portion of cells that when they've undergone this, supposedly NHEJ repair, they have all sorts of all sorts of kind of long range effects. You have like inversions, meaning, you know, regions of the, you know, that DNA in that region kind of switched around, swapped. You have big chunks missing, you know, that are on the order of kilobases long. And this could, you know, lead to kind of far off effects, because obviously, you know, if you're dealing with kilobases of, you know, changes and not just 10 to 50 bases of changes, then you might be interfering with genes that are surrounding the gene that you're interested in targeting, right? So let me, with that, switch over to the paper itself. So share my screen. This is the right paper. Let me stop sharing briefly. Let me share this again. Oops. There we go. Wrong one. Share screen. All right. So here's the paper itself. So it is the repair of double-strand breaks induced by CRISPR-Cas9 leads to large deletions and complex rearrangements by Kosicki, Tomberg, and Bradley over at Wellcome Sanger Institute, Hingston, UK. So over in your neck of the woods, Steve. So basically, in a nutshell, I'm just going to kind of summarize what they're working with here. So what they're looking at in this paper is, so what they mentioned right here off the bat, you know, it's a very, I think it's a pretty tight paper. They say that the exploration of Cas9-induced genetic alterations has been limited to the immediate vicinity of the target site, which is what I mentioned, you know, a few tens of bases upstream and downstream of where the Cas9 cuts, and distal off-target sequences. So, you know, other chromosomes way out somewhere else in the genome. What they're now looking at is around the target site, and they're noticing these large deletions around the target site, which was missed by, I guess, somewhat surprisingly, people, you know, have not been looking in this sort of intermediate zone for whatever reason. They haven't really been thoroughly sequencing, you know, a few kilobases up and downstream, and you know, that's probably because people were mainly, interested in things that are way off target, genes that are far away, and if they assume that, you know, the cells you got are broken, you know, in the location, then everything, why should there be any changes a few kilobases away? Turns out that that was a false assumption, that there are such changes. So they're looking at three types of cells. In their paper, they're looking at mouse embryonic stem cells. They're looking at mouse hematopoietic progenitor cells. So these are normal cells. So these are cells that are not cancerous. And then they were looking at a human differentiated cell line. And the system that they're looking with is the gene that they picked is this locus called PIG-A. And PIG-A, and I'm just going to scroll to the first figure here. PIG-A encodes phosphatidylinositol-glycan class A gene, and it's an X-linked gene. So that means if you're using male cells, then it's hemizygous, meaning that, you know, if you don't have to worry about a second copy kind of messing around with your results here. You only have one copy, and even with female cells, because of the formation of bar bodies, meaning that, you know, you have one chromosome that's inactivated, you know, you still get this hemizygosity. So their only worry is with this, you know, one locus here that they're gonna take a look at. And there are systems that have been developed to basically, you know, essentially sense if the PIG-A locus is knocked out. This gene basically attaches GPI anchors to cells, and then there's a label that you could use. I believe it's a bacterial protein, and the acronym they use is called FLAIR. I don't know how I pronounced it. FLAIR, F-L-A-E-R. And it's basically a fluorescence stain. And this fluorescence stain allows you to use FACS sorting, so fluorescence activated cell sorting, to look at cells or find cells, enriched cells that have been knocked out for the functionality of the PIG-A locus. And that's what you see here. So they use this system called piggyback, a transposon-based system, to get their Cas9 and their guide RNA. And this stuff here is just essentially the vectors they use. And once they get the system in, they can then cut it out. You know, before we get into this, you know, my question is, you know, I read this paper, I've probably read through it about 2 times, so I'm not a PacBio systems expert. So if anybody here has used the PacBio system, you know, for sequencing, please chime in, you know, and help me out here. But basically, you know, my understanding is, you know, I've read a number of CRISPR-Cas9 papers. So they use this piggyback system that's a transposon that inserts in a number of different locations in the genome. They mention in the paper this, you know, the particular kind of sequence where it sort of prefers to lodge into. And then you use this PBase enzyme to get rid of it. And so what they do is they have these mouse embryonic stem cells, PigA positive. They knock it out. They use liposuction, I guess, to get in. They select, you know, using antibiotics, and then they find cells that are fluorescently labeled or not labeled. So I guess this is pyromycin, and the FACS sorting is done for PigA minus. So cells that are knocked out are not going to be fluorescent, right? So here are your fluorescence kind of scatter plots. SSCA is basically a side scatter. Kind of a general scatter for cell size and granularity, and PigA is your flare, you know, your fluorescence. So this cluster of dots here is your negative control, so they're all expressing PigA, so they all have a GPI anchor that's labeled. And then you have these numbers here, number 10, number 56, are all different guide RNAs. So here is one particular type of experiment that they have. This is the locus PigA. These blocks here are exons. So we all recall there's exons and introns in eukaryotic cells that are spliced together to make a functional gene. And this is a guide of size, so 5 kilobases. And these are percentage of PigA deficient cells. And here you have this little plot here for the percentage of cells depending on where you have the guide RNA knocking things out. So you notice even in intronic sequences, it's rather low, but you notice so intronic greater than 2 kb from the exon, intronic, you know, close to the exon, and exonic, so obviously knocking out things in the exon is pretty bad, meaning that the cells are at a high efficiency, you know, they're PigA deficient. So you can see exonic 56, very little cells have this flare, but you still have this, you know, even throughout introns all over the place, even quite a distance away from the, you know, exons, like right this intron here. You know, and they mentioned in the paper, they looked at, you know, what sort of changes have occurred. So they're not, it's not a subtle change in just an intronic splice site, right? So this is, you're really missing like chunks of DNA that are going well into the exons themselves. And that's the issue they raised in the paper. So you still have this kind of high level of knocking out that's close to 20%, you know, even in intronic sequences. Now, one thing I have to mention, and one question I do have is how long is this Cas9 guided RNA system being expressed in their system, right? So again, they look at 3 cells. So they look at two mouse cells that are embryonic cells, embryonic stem cells, and hematopoietic progenitor cells. So basically adult stem cells. And then later on, we'll take a look at a human differentiated cell line. Bottom line is they get the similar results for all of these. I'm not sure, so they, you know, they do 14-day selection, but I do know that if you express Cas9 and CRISPR for a while, I don't know what a while exactly is, but depending on the cell line, the papers are, you know, people will have published that there's toxicity involved. So I take that to mean that there is off-target cutting if you have CRISPR-Cas9 being expressed for a while. So that's kind of one question I had here, was exactly how long was this being expressed? It's probably somewhere in there in the paper. And the other issue that I'm gonna, you know, mention to people that, you know, if they're just coming to this paper for the first time, and I'm gonna stop sharing, is that this paper is, to the best of my knowledge, after I've read it, they're not looking at homology-mediated repair, right? So this is just purely cut the DNA and break it, right? So what, you know, my other question is, and I'm kind of skipping ahead to the end, is would this be a similar issue if you had homology-mediated repair taking place? Because my understanding is for many gene therapies, I guess for some I can imagine you want to knock things out, right? So one example I can imagine, I can think of is for anti-HIV therapy is knocking out the CCR5 locus, right? So that would be just a straight breaking, you know, your CCR5 gene or not targeting an exon. Hopefully it doesn't affect things up and downstream, which this paper suggests it might. But what about in other gene therapies where you're actually trying to fix a broken gene, you know, like let's say a mutation in cystic fibrosis, right? In that case, you have a template. Would you have the same issue with these gross deletions and inversions if you had a template that's template-mediated repair taking place? Is that still an issue? You know, that might be a good follow-up paper to see what happens in the context of homology-mediated repair. But anyway, so that's their first figure. I'm going to pause here before we kind of look at the sequencing data, which, you know, basically it just shows you all of the changes that are occurring. Take a little pause. Breathe a little air. I wish I had a cup of coffee, but I'm in the lab, so I can't drink any coffee. Okay, so let's go back to the paper itself. Okay, so I'm going to scroll through this. And here we have our, maybe we can get some of the other guys to chime in here. So we have figure 2, and this is the PIG-A locus. So I'm just gonna kind of lay this out. This is a negative control. So again, I'm not an expert in genome sequencing and genome sequence analysis. They have read depth here, which is the amount of reads that were done. So in order to get really kind of detailed analysis, you basically have to repeat like individual reads, and it looks like they've repeated quite a lot. So, you know, here we have for, you know, for unsorted cells and PIG-A negative cells and PIG-A positive cells. So unsorted meaning they haven't run it through a fluorescence activated cell sorter. So this is just after they transfect the cells, you know, and you have a mixture of cells that are knocked out, not knocked out. You're basically a pool of cells. PIG-A negative are cells that you've clearly knocked out the PIG-A gene, and PIG-A positive cells are cells that are still expressing PIG-A and are then fluorescently labeled. And these, so those are, so this is in different colors, color coordinated. NC is negative control, so they use the guide RNA that's not specific for anything. This gray bar going through is the exon number 2, and these dashed lines, these lines that are going vertically, I believe are the guide RNA cut sites that are a few bases away from the protospacer adjacent motif. And then this here on the right is kind of a zooming in of this gray bar here. So this is 100 bases and this is 5,500 bases. And these are regions that have been, I guess, sequenced. And this is the amount of times that they've been able to, the PAC biosystem is able to essentially get a read or get a correct read at these locations. And if DNA is missing, then this thing dips down, is my understanding, right? So you basically see big swaths of DNA that are kind of missing from regions that are both proximal and distal to this, the cut site. So these are unsorted cells, and clearly this is going to be much clearer, this canyon here, than the unsorted cells, because here you have, masking this, you have a whole pool of cells that are not sorted, that are basically the PIG-A positive cells. Interestingly, even PIG-A positive cells, you've got stuff that appears to be areas that are missing. It might be chunks that are, even though they're PIG-A positive, they're probably maybe a chunk missing from an intron. I don't know. That's a possibility. So these are read depths. So they've done a lot more read depths for the PIG-A negative cells, I guess, just for more thoroughness. Again, somebody with more background in the PAC biosystem would explain why the read depth is much more depthy for these PIG-A negative cells, so 2,000, 4,000, 5,000 versus several hundred, but nevertheless, several hundred read depths is pretty thorough. So you get quite a lot of these changes occurring. And then, here they have a diagram of the sequenced allele and a PIGA reference allele that they basically used, and some of the changes that they that they've plotted that may occur when they actually, so there's a lot of supplemental data. So a lot of the data in the paper that we're actually not covering here is in the supplementals, the types of changes that the sequence data pulled out. So now we're not showing you the sequence information here. So obviously here you're just looking at read depth and how much, you know, and how much sequence data they can actually come out. So clearly what they're looking at here is, you know, chunks of DNA missing, but it's not just chunks of DNA that's missing, it's also DNA that is present but switched around. So those are inversions and changes of that nature. So I'm going to take a pause here. And anybody wants to chime in, feel free to chime in at the moment.
Well, I haven't read this paper yet, but in 2016 there was a paper published in Science Reports, and according to what I have read, apparently they did a whole exome sequencing of knock-in mice that had been generated with CRISPR-Cas, and while they did observe some indels in that study, they, as far as I understand, didn't find any major rearrangements, no inversions or big deletions or that type of stuff. But again, I didn't read the paper, so I can only, you know, say very briefly. I scrolled a bit through it, but didn't read in detail. But that seems to be a little bit in contrast to what we hear now. Though these were knock-in mice, so they did use homology-directed repair.
You mean in the other paper?
Yes, yeah, in
okay.
the 2016 paper.
So this would be a great contrast. So that suggests that it might not be an issue if you do HDR, right?
Yeah, and of course you could use HDR as well if you want to make a knockout. Right,
of course. So that may be a workaround if that's, you know, if that's your worry, then using a template might suppress a lot of what we're seeing here. Basically, yes. It is what that other paper suggests. So that's, yeah.
There are also small molecules in existence that can suppress non-homology, non-homology directed enjoining, and there are also those that stimulate homology directed repair. So probably you could, you know, use those as well to suppress large deletions.
And this is also, it strikes me, this is kind of an issue, more of an issue if you're going to do this in vivo in an organism, because a lot of gene therapies, you know, a lot of therapies that are being developed are basically you take cells out of a patient's autologous cells, you manipulate them in vitro, you screen them, you select them, and then you reintroduce them back to the patient. So if you have this ability to sequence them, right? If you enrich for a population, you know, from a cell, and then you can sequence that population and then select for ones that don't have any kind of major rearrangements, then, you know, that to me would seem to be, you know, a pretty straightforward, you know, checkpoint to put into your protocol if you're going to be doing this in human, you know, trials or patients, right? I mean, if you're already taking cells out of a patient, basically employ the same PacBio approach that they used, you know, the pre-screen cells. Could be more of an issue if you're doing this in vivo, because then you can't really be pre-screening cells. But like you mentioned, Sven, you could basically maybe use some sort of you know, homology-mediated repair system, you know, if you're going to, even if you're doing disruptions, to kind of squelch, you know, these problems that might happen with excessive NHEJ. Or something else, or a drug.
also worth mentioning at this point that CRISPR Cas9, which this paper is about, that is only one of the various types of CRISPRs, isn't it?
Mm-hmm.
We've got other, I can't think off the top of my head which ones, but there are some that appear to have more sort of utility in mammalian cells. Some are more useful in different types of species. So again, it'd be interesting to see as a follow-up potentially what some of these other types of CRISPRs are like in regards to these mutations.
Right. Right. So so there is still figure. We went through figure 1. We went through figure 2. There is still figure 3 and figure 4. I don't know how much point there is to go through figure 3 and 4 since they are virtual. Well, I don't want to say identical to figure 2. But basically, it's the same approach they've employed, but figure 3 looks at autosomal CD. They basically look at another locus in mouse embryonic cells, and then they look at PIG-A loss in a human, I think a retinal cell type, an RPE1 cell. And using the same piggyback transposon system that we looked at figure 2, and they get the same thing. They get insertions, deletions that are pretty big, that are, you know, both 5' and 3'. And I think, you know, what else was I going to say before this phone started ringing in my background here and I started to lose my train of thought? We are in a lab, so apologies for that. So you mentioned, Steve, that, you know, there are these other Cas systems. There is the system that you make staggered cuts, right, versus a single cut. And I think the staggered cuts, that is, you know, it's less likely to do NHEJ, more preferable for homology-mediated, homology-mediated repair. There's also other, you know, programmable endonucleases. Now, you would assume that what's the difference if you use Cas9 CRISPR, or if you use TALENs, or you use, you know, or you use zinc finger endonucleases? Programmable is programmable. Double-stranded break is a double-stranded break. I don't know. I mean, these are protein complexes, right? And they sit at a location, they go someplace, and they make a cut, and they might potentially interfere with other enzymes that are in the locus, in the location. So, you know, it'd be interesting to see, you know, if you basically looked at other, you know, other programmable endonucleases, whether or not this same issue arises. You know, that might be a little excessive, but, you know, again, if you're doing these for clinical trials, then, you know, and this paper certainly raised this major issue of these kind of, you know, major changes that can happen several kilobases upstream and downstream, then it might be well worth looking at, you know, all the other ones, repeating this, you know, this data with all the other ones, revisiting the paper that Sven mentioned, where they looked at, you know, homology-mediated repair and didn't see these types of major changes, and see whether or not, you know, maybe repeat some of these experiments with the same guide RNAs and use templates and other methods to suppress NHEJ, and to see whether or not you get these types of changes going away, for example, or you get a major suppression. You know, I think this paper, you know, lays the groundwork for, you know, future papers that would maybe address this situation.
Oh, and just whilst I remember, it was CRISPR CPF1 that was more suited to mammalian cells, which they demonstrated last year. I don't know if you guys know that one. CPF1 is the particular type of CRISPR that seems to be more accurate for mammalian cells. Obviously, CRISPR-Cas9 has enjoyed the most notoriety. But yeah, there is quite a lot of different ones out there.
And yeah, you know, homology-mediated repair and NHEJ have been studied for a long time. You know, I haven't dug through the literature, and you know, I would be surprised if people in general, well, I mean, I think they do mention in this paper here using a specific endonuclease. So let's see. So this is going into the first page of the paper we just, you know, mostly looked at, and that's, they mention in their introductions, lesions not contiguous with the cleavage site, such as those reported in yeast upon ICE1 nuclease cutting, which is a endonuclease that recognizes a long stretch of nucleotides that's, you know, used in yeast, would also be missed by such short range assessments. So, you know, these sort of long range changes that occur within a cut site have been noted before with other endonucleases. You know, I guess the question is really, why haven't people been looking at this with CRISPR-Cas9 from the get-go, right? Since this isn't, you know, this isn't inherent, really. You know, I don't want, this really isn't, doesn't seem to be inherent to CRISPR-Cas9. It's inherent to double-stranded breaks in a wide variety of eukaryotic cells, and potentially due to the way the NHEJ system works, right? So this, you know, this appears to be something that's kind of baked in, you know, into the operating system of most eukaryotic cells when they see a double-stranded break and they resort to NHEJ, perhaps. So I don't think
kind of leads to the question is, I mean, I'm not familiar with that literature, so I don't really know, but, you know, have other people not seen these large scale rearrangements, deletions happening in people using talons or zinc fingers or other things? Because, I mean, people have used talons, for example, a lot as an experimental tool in, for example, frogs. People who do research on frogs, genetics, and so on, have used this for like 10 years already also, that they have done like thousands of studies knocking out all types of genes and so on. So have those people not observed any large scale rearrangements?
Yeah, I don't know either. We'd have to take a look in the literature and see, and it depends on how they you know, how the studies were done, right? If they knocked out a gene and they selected, you know, they wanted, if they already pre-selected for a cell in vitro and they were looking for a cell that had a particular region knocked out, and then if the other cells didn't fit that criteria and were just thrown in the garbage, then, you know, that could have just been somehow screened out, and they just weren't interested in following up on the other cells, right? Because it's, you know, you get a lot of, I mean, this is kind of an interesting, I guess, more also a scientific experiment, right? Because you do this kind of, you do these, the same guide RNA could generate all these potential differences. You know, so this is almost like a screen for what happens during NHEJ, right? So all the different things that can happen, you know, inversions, translocations, deletions, insertions, right? Like, why does all of this happen, right? What are all the different, you know, are there all distinct pathways that lead to all of these distinct changes occurring, right? Because you kind of get this gamish of things happening, but can you untangle all of this? Are they all separate subsystems that are, you know, vying for this repair at this locus? And, you know, can they all be kind of teased apart individually, right? Is there a different mechanism for a 2 kilobase insertion versus a 2 kilobase deletion versus a 2 kilobase inversion versus a 2 kilobase translocation? I don't think there's one enzyme that's doing all of that. It's, you know, there's a lot of things that are happening, and I think people are still trying to figure out, you know, all the different components of NHEJ and homology-mediated repair. And I'm just also thinking about it from a scientific perspective. Can this be used as a tool to basically screen for, you know, isolating different types of, you know, events that happen in an NHEJ, right? I mean, can you have a whole collection of different mutant cells, and each of them is defective in one form of NHEJ, right? Are there cell types that, when they do NHEJ, never do deletions? I don't know, right? I mean, I'm just now moving away from the engineering perspective and more into the scientific perspective. You know, just as a tool for studying this process of what's happening at that locus, right? Because I think a lot of times people might miss this kind of stuff because, you know, they're focused on the end goal, which is admirable, which is you're developing a gene therapy. You want to do your modification, you get the modification, and all the other 10,000 things that come up while you're doing the modification to that one cell, those end up in the garbage can because you're just not interested in that. Why would you be? You're interested in making a therapy, right? All the things that supposedly don't work, why do you even care about that? Especially if you're going to be screening for a cell. But I don't know, maybe from a scientific perspective, it's interesting to kind of go rooting around in a garbage can and see what, you know, see what that might tell you about the underlying mechanism of what's happening when the cell is trying to repair itself, and why does the cell choose these different pathways? You know, these wildly different, you know, ways of kind of doing the repair. You know, people just say that NHEJ is more error prone. I guess whatever that means, right? It's more error prone. But if it's more error prone, then you wouldn't in general see this wide range of changes. You would see, you know, maybe a few tens of bases here or there, you know, maybe the polymerase adds a little more, maybe it adds a little less. You know, maybe the ligase is a little too, you know, I mean, not the ligase, you know, the nucleases just cleave away a little too much, but why do you have all these other changes occurring, right? And can they be individually suppressed? And they can, if we just basically throw in templates and shunt things towards homology-mediated repair, but are there more subtle ways to suppress what's going on here? My 2 cents is that, you know, this is a good paper. It basically tells researchers, especially, you know, in the gene therapy field, what to look for as potential problem. But I think these problems are solvable, you know, based on what this paper has shown and based on, you know, other papers in the past. That you know, if you're doing most gene therapies, if you're taking cells out and doing them in vitro modifications and having autologous cells, then it's just a matter of adding in another checkpoint or two to screen your cells to make sure they don't have these changes before you put them back into a patient. Mm-hmm. If it's in vitro, if it's in vivo in the patient, then you've got more hurdles to overcome, and that's probably, you know, we're now going to have to think about better ways to suppress NHEJ, perhaps, right? But I think these are all, you know, overcomable problems, I think. And I think that even though this paper looked only at CRISPR-Cas9, I would be surprised if these problems aren't inherent in all the other programmable endonucleases, especially since it's been noted in ICE1 nucleases in yeast, you know, which is a non-programmable endonuclease that's a native endonuclease in yeast. I mean, this is probably, you know, a generic issue for eukaryotic cells.
And it's also already known that there are at least 2 pathways to non-homologous end joining, the canonical and the alternative pathway. And the canonical apparently leads to no or very small deletions or indels, while the alternative pathway leads to indel generation. So, you know, you're perfectly right, and maybe we don't even know, maybe there might be even more than 2 pathways there, and some of them could be more precise and lead to smaller changes, and others could be less precise and lead to bigger deletions, bigger rearrangements, and so on.
Yeah, and even if this system is, you know, even if you get these major changes that happen here, you know, if you're targeting a gene, if your goal is to knock out some kind of gene function, and I mentioned CCR5 as one example potentially knocking out a gene which is, already being utilized as a potential therapy for curing people of AIDS and HIV infection. If I don't know what's around the CCR5 locus or how big the CCR5, how spread out the exons are and introns, but if there's nothing supposedly important around that whole area, then you might be able to just get away with, targeting it and getting in some kind of wacky inversion and 3 kilobases missing, and hey, it's fine, right? In some contexts it might not be fine, and that's the issue. But in some cases, depending on what's around the area, and how it's interacting with other regions of the chromatin in that area, it might be a problem. So even with these issues, it's also very context dependent. How big of a problem having a big chunk of DNA missing when you're only expecting, a few tens of bases missing, right? So it depends. Fortunately, we live in a time right now when most genomes are being sequenced and are being easily sequenced. So it's not a mystery to basically see what sort of changes may have occurred. We don't have to scratch our heads. We can actually go and look and see whether or not, our programmable endonuclease of choice has done anything funky. I think it's heartening that we don't have any of these, or very little of these long distance effects happening. To me, it seems to be a lot easier to search within the vicinity of the gene to see if there's any major changes. If that's the only thing we have to worry about, then I think that's not a huge worry. There's constraints, and we've got the technology to look and see what's going on. I think the fact that the paper raised this, that we should be looking when we do this, I think this is a positive, and will make actually gene therapies even safer, if they implement some safeguards to just check and make sure that such changes haven't occurred.
Yeah, so basically here is the take home is essentially this is merely a bump in the road really here, and it's all about engineering as much safety as possible, like George Church talks about engineering safety, I should say. So that's good news, right? That means we're on track for woolly mammoths and dinosaurs still.
Yeah, well, what this paper nicely does is to tell people, warn people what to look for if things go wrong and where to look for the things that might go wrong. It's a lot worse not knowing where to even look, right?
Yeah.
So not having to worry about the rest of the genome and kind of focusing your attention to a region that's a bit more constrained, I think is a plus in my book. Yeah, you have to, there's more work that needs to be done now if you're going to be doing a gene therapy to make sure things haven't gone, awry, but such as safety, right? You have to.
Oliver, have you checked out the Cas9 clinical trials that they mentioned here in the paper?
I haven't checked those out. I was going to, the ones that they mentioned in the first page. The first, consequently, Cas9 is reasonably specific, and the first approved trials using Cas9 edited cells are underway. Yeah, so they mentioned like 1, 2, 3, 4, 5, 6. So they said Cas9 edited cells. So that tells me that they selected these cells, in vitro. What were they looking for when they selected these cells? I don't know. I haven't pulled up those trials, and it'd be interesting for these guys here to pull out those cells and see if any major changes have occurred. Now, of course, it's going to depend on what they knocked out and where, what region of the, genome that they knocked out. If it's a gene that's surrounded by areas that are normally not transcribed, then there might not be such of a worry, right? If they get like several kilobases missing. If it's a gene that's proximal to a proto-oncogene, then they might want to be worried, right? or a tumor suppressor gene. So yeah, it'd be interesting to take a look at these trials and see what's what exactly they altered, or whether even they did homology mediated, you know, and repair. I don't know. They just say Cas9 edited cells, so it could be anything. But if anybody wants to look up those clinical trials, the numbers are there. Go to clinicaltrials.gov, and I'm sure you could, you know, sure they'll tell you the cells that they used, although probably not the relevant sequence information. So, unless anybody wants to do figures 3 and 4, which are a bit repetitious, I think I want to skip those because we've gotten to the, I think, to the heart of what this paper has been talking about. And like I said, they looked at 3 cell types, and their stress is that they're not tumor cell lines, they're not cancerous, right? There's 2 mouse cells that are an embryonic stem cell and a somatic adult stem cell, and also a human differentiated cell line that's a retinal, I believe, a retinal, some sort of retinal cell. I have to take a look in the paper. So, you know, as far as tissue culture is concerned, you know, these are about as normal as you can sort of hope to get, right? So yeah. So do we have any questions from anybody on this paper?
Nothing from social media. They're all very quiet this week. Don't know what's got into them. Perhaps they're just that fascinated. But no, there doesn't seem to be any questions. But yeah, from this paper, my take home is, yeah, it is a bit of a challenge, but you know, nothing that I don't think science can't overcome, right?
Yeah, and like I said, you know, I like this paper because it sets constraints to, you know, it's like that quote from our philosopher warrior Donald Rumsfeld, right? What's the? I want to make sure that I get this. Anybody remember Donald Rumsfeld? Rumsfeld.
I don't know. I think it's a bit before most of our time, or possibly cultural reference or country, or that's the problem when you got an international team, Oliver. The pop culture references are all they don't necessarily, what makes sense in one country doesn't necessarily make sense in another.
So this is during a Pentagon news briefing in 2002. Rumsfeld stated that reports that say that something hasn't happened are always interesting to me because, as we know, there are known knowns. There are things we know we know. We also know there are unknown unknowns. That is to say, we know there are some things we do not know, but there are also unknown unknowns, the ones that we don't know we don't know. And one looks throughout the history of our country and other free countries, it is the latter category that tends to be the difficult one. So what I like about this paper is that it basically gets rid of some of these unknown unknowns. And yeah.
It's a bit like Aubrey's joke, isn't it, about metabolism, about there's a lot of things we don't know about metabolism, and that's excluding the things that we don't know about.
Yeah.
So, you know, it's the ultimate unedited spaghetti code, as he calls it, doesn't he? Yep. But yeah, I saw the conference video today from that we've just did in New York with Aubrey's bit, and he had to throw the joke in about metabolism. So it's a classic, isn't it?
Always.
It's a standard, but it's funny. But yeah, you know, at least eliminating as many unknowns as possible is a positive move, especially when we're playing about with the genome, right? Otherwise, you know what happens? Jurassic Park happens.
Yeah, and from my watching of movies, it happens really fast and really easily.
It does, yeah, and it's like, you know, and then people say, "You scientists," what was the line? You were so obsessed, you were so preoccupied with whether you could do it, you never asked if you should do it.
I know.
So we better be on our best behavior, right? Because we've got to make sure we absolutely get it right. And I mean, joking aside, medicine is a serious business, and we've got to, and especially what we're doing we've got to make sure safety is absolutely paramount. We don't want anybody turning into the Mothman or becoming a human man-bear-pig hybrid.
Or most likely just die of a horrible tumor.
Because man-bear-pig, I mean, it's real, right? I mean, it's definitely a possibility, right, Oliver?
Well, as a scientist, I can tell you that it is a possibility.
Man-bear-pig, you see, there you go.
I'm not gonna quantify that possibility, but I will say it is a possibility.
Yeah, there you go. You heard it officially from a genetic engineer. Man-bear-pig is a potential possibility.
Yeah.
They
Potential.
They weren't kidding around in South Park at all.
No, they weren't.
Neither are we.
Okay, so I'm actually, I'm happy with this paper. I'm happy with the issues that it raises, and it's great that it also points people to where to look. Right? There's always people mentioning a problem and then not coming up with a solution, but this paper does mention a problem and does point out some solutions. And I think there might be other solutions as well that we raised on this broadcast here, based on earlier papers that were published. And it's definitely like when you hear like rumors and hype coming, from kind of sideways, it's like, the paper just came out that crushes CRISPR-Cas9 and says it's over. I'm like, no, that's not what this paper's about.
Yeah, and those stories have been doing the rounds.
No.
Just the same as the last lot, where that very flawed experiment with the 3 mice, everybody was on the bandwagon. It's all over. We're never going to be able to do it. I'm like, guys, it's one paper, right? Look at it as a challenge. But I suppose sensationalism is what gets clicks and gets people to visit websites, isn't it?
Yeah. And, again, just like we mentioned with all these changes that occur with NHEJ, I mean, there's still massive amounts of processes happening in cells that we have no clue, the unknown unknowns of what's going on. We try our best to kind of, to engineer and hope that everything else is a distinct, orthologous black box that's not going to interfere with what we're doing here. But that assumption, in many cases is flat out wrong. And, that's when the science intrudes into the engineering, and that's always going to be the case, right? And but what I like so far, what I like for all the papers that have come out so far, on CRISPR, Cas9, and the other programmable endonuclease stuff, and NHEJ and HMR, and methods that people are coming up with to make the systems more precise, is that that we are seemingly on the right track, that that we are making the system more precise. It's never going to be perfect, so just take the word perfect and 100% and throw it out the window. It doesn't exist in engineering or science. But you want to get to acceptable levels of risk, whatever number you set, and that's the best you can do. And I think we're really, really far along in that direction, to acceptable risk levels, and that's really the best that we can hope for.
Yeah, I mean, I've said many times, all medicine is risky. Even taking an aspirin, there is an element of risk to it. So when you're doing complicated things like, well, genetic editing and engineering, sure, yeah, there's risks. So the more we can build safety in, the better. But of course, there comes a point where the risk versus the reward is worth it. And if we can do things like eliminate things like Duchenne's muscular dystrophy, age-related diseases, hereditary conditions, there are quite a few inherited conditions that people suffer from. If we can rid the world of those things, then to be honest with you, I say we, the risk is worth it as long as we proceed with engineering safety.
Yeah, and one thing, one thing people always neglect when they talk about risk is the risk of not doing anything, right?
Yeah.
I mean, so you have to factor that in, right? If there's a negligible risk that somebody might, get a cancer or die from a particular therapy, well, don't ignore the other half of the equation, which is the known risk of thousands of people dying if you don't attempt it, right? And a lot of times, the naysayers neglect that completely. That's left out of the equation, and that's wrong, right? You have to also factor in the deaths that will occur and are occurring if you choose to do nothing, right? So and that always irritates me. That always makes me very, very angry when those numbers are basically hidden behind the curtain, right? So those numbers should be front and center as well.
Well, I mean, it's one of those things that for some reason people feel that if they do something, then and it results in a bad effect, then they are responsible.
Of course, it's responsibility, right?
But
because
if you don't do anything and things go wrong, then it is not your fault, right? I mean, there is a famous thing of that philosopher, what was it? Was it Descartes or who was it again? Who basically said, okay, imagine you see someone running away and, he says, I need to hide because there is a murderer running behind me. And then the murderer comes, will you tell him in what direction that the victim was running, that he's trying to kill? And Descartes comes to the conclusion, well, I mean, if you will point to the right if you will point the killer to the wrong direction, but the victim changed his idea after you saw him running in one direction and returned into the other direction, then you are responsible because you, by accident, pointed the murderer in the right direction. Well, if you were honest and you said, oh, he's running in that direction, then even though the murderer has a much higher probability of killing the victim, it's not your fault. You didn't do anything wrong, right? That's Descartes. He basically came to the conclusion you should never lie in any case, right? And always say the truth, even if there is the highest probability that will lead to the person being killed, because you just absolve yourself of any blame in the matter. And obviously, I don't agree with that type of approach at all. I take a much more, rational type of approach where I say, well, what matters is the outcome in a sense. If you have doing nothing is a higher risk than doing something, then you should do something.
I'm going to be a cynic here and stick my neck out and say that even in a non-religious, classically based liberal society, there's still one place for God left, and that is to deflect blame, right?
Mm-hmm.
So if we, so that comes to personal responsibility, right? If you're going to be a truly non-religious, right, classical society, then you have to accept personal responsibility and personal blame. But when it comes to these types of therapies, nobody wants to do that. We'd rather pawn off the victims on a deity, right? Just don't blame me. That was God's will. But if something goes right, well, then hey, I'll take the credit for it.
I mean, there is also the reticence in medicine as well to do no harm, which is obviously a core part of, practicing medicine. So, there is a certain amount of hesitation. And one thing I always think of is individual rights. If for instance, you're suffering from a disease or something, and you personally accept and understand the risks, then I'm all in favor of doing that. The right to try is something that really needs to be amped up.
Well, wasn't there a very, very controversial case in the UK a couple of years ago, right? When parents raised money for a child to undergo a very, probably wouldn't work, but some sort of mitochondrial therapy for their baby, who basically was going to die. And the UK courts basically said that it was illegal for them to even attempt to do a therapy, and that the baby's best interest would be to flat out die.
Yeah, the Charlie Gard case.
Yeah, which
Yeah.
I thought was particularly outrageous, but hey, what do I know? I'm not a barrister or a judge.
Yeah, that was a very divisive issue. But yeah, I definitely support right to try, you know, within reason, and, you know, obviously safety. Safety is always a concern, but it shouldn't reach the point where it paralyzes, you know, progress. I guess that's what I'm driving at here.
Yeah. Well, yeah. I mean, it'll be interesting to see how all of this develops, right? Because it comes down to who takes the blame, you know, around, you know, if it's, yeah, it's we don't have time to go into this. Basically, this is the whole nother discussion. But perhaps we will have a discussion of sorts related to, you know, how clinical trials are arranged. And I'd love to, you know, talk more about this because, you know, at least the biggest market for most drug companies is for a variety of economic reasons, the United States, you know, because it's sort of the Wild West when it comes to drug prices. But I'd love to see how other countries are doing things. I'd love to see, you know, how, you know, what's the best way to get these therapeutics, you know, to get therapeutics out? What's the best way to weigh the inevitable people dying when they, you know, undergo untested treatments? I mean, because let's face it, right? It's untested, so there is a risk that something horrible is going to happen.
Yeah.
But what's the, you know, what's the best way? What's, you know, is there an ideal system that could be in place that basically kind of, you know, weighs all of these factors and in the end, you know, gives you the best result? And by best result, I mean, you know, sort of getting back to, you know, John Stuart Mill, right? The greatest good for the most people, right? That's really that's the equation you want to go for. You don't want to go for nobody dead because, you know, somebody obviously is going to be dead. So what really the system is geared now is to have nobody dead that can be assigned a particular blame to an individual, right? That's kind of broken because clearly if all the responsibility for somebody dropping dead was put on my shoulders, then I would halt all trials, right? Because, you know, who wants that responsibility? So is there a system that can basically, you know, weigh the greater good for the greatest amount of people when it comes to trials? And, you know, obviously we can talk about government, all that, but just within the narrow spectrum of therapies and therapeutics and saving lives and getting medicines that, you know, tested, you know, what is the best system? And I don't know, I'm sure people must have done comparative analyses, right? If they have, I haven't really read them, or I haven't read the magnum opus of comparative analysis of you know, of drug trials and medicine. But there's got to be something like that out there. If not, I know people have been thinking about this, been doing some, being really studious about it. So maybe we should have those people, you know, on air, and maybe we should do some sort of podcast, because I would be interested in hearing all of the alternatives and really vetted alternatives too. I know there's, you know, you can go from one extreme to the other. Obviously, when it comes to a personal right and personal individual right, I can test whatever the hell I want on myself. Well, maybe not. I mean, the federal government has its own quirks about me taking certain things that are mind altering. But that being said, unless, you know, if nobody knows about it, you're pretty much free to do whatever you want. That's safe to say. But how do you get this information out, right? Because you want other people to trust it. You want it to be replicable. You want it to be, you know, used by as many people as possible. So really, what's the best system out there, you know? Yeah, that'd be a great discussion to have.
Yeah, and it's going to become more and more relevant as well, as especially in regards to the therapies for aging, because they're literally now starting to arrive. You know, we've got Senolytics that are already entering human trials with Unity, for example. You've got Clearer Biotech, you've got Antoxerine, and quite a few other companies now that are hot on their heels. This sort of conversation is going to start occurring more and more often as these rejuvenation technologies arrive. So it's better really to have the conversation now than wait. Otherwise, society is going to be caught, you know, on the back foot, as it were, because these things are coming, and people are going, "Oh, yeah, it'll never happen, not in my lifetime." It's like, what do you mean it'll never happen in your lifetime? These things are already in human trials. They're coming. So it's best that we have this conversation now, and we work out the best path ahead. So yeah, definitely I would be up for a special journal club to discuss things like that.
And if you look at, for example, the rapalogs, I mean, it has been known for quite some time that they extend lifespan quite dramatically in mice and so on, but still it's very hard to obtain it. You can't get it for personal use, and just it takes time for sure.
Yeah, so there's a whole can of worms there. We've got enough for a whole show's worth. Perhaps we should get a bunch of people who are specialists in policy and such like, and other luminaries, and let's do a special journal club.
And all these technologies are interrelated. So right now, if you don't like the way your country's doing its drug policies, well, it's pretty cheap, fairly cheap these days to travel halfway around the world and go someplace else.
Yeah.
So, price of a plane ticket is in many cases way cheaper than many drugs, right? So what's the best way for the most people to be as healthy as possible? That's the bottom line, right?
Yeah.
But obviously, I mean, it's going to be medical tourism when it's finally out there, when you have clinical products. If it's just one small island that allows you to have rejuvenation therapies, everyone is going to fly there. So that's
very shortly going to be the richest island in the world as well, and the most popular for plane visits, flights and holidays and medical vacations. But yeah, it's an interesting subject in itself.
And the oldest incoming population and the youngest outgoing population.
Exactly. They go in old and knackered, Oliver, and they come out shiny new. We're not so much about making new people, are we? We like to recycle the ones we've got, which is something that I often joke about.
And hopefully this will be a global reality and not like that cheesy 70s show, The Fantasy Island, right?
Oh dear, not that. Oh
anyway,
dear.
Maybe I just jumped the shark here. Okay.
Yeah, oh dear. Yeah, you always have to end on a down note, don't you?
Well, that was kind of a fun show. No, it's awful. I don't remember. I was a wee little kid when the show.
Oh, I don't know. I'm gonna put that along with a lot of the shows from the 70s, but not, however, that 70s show, which wasn't actually in the 70s, which was pretty funny.
And I should just correct myself. Actually, it was of course not Descartes, but Kant, who came up with that idea.
Well, it's too late to edit that now. I'm not doing it, and I'm not doing it in the dub. So there we go. We've got a possible subject for next time. Maybe we'll see how it goes. And thanks for going through that today. Hopefully, it's allayed some fears and concerns. But we always try and give a sort of balanced answer. We don't want to be blinded by optimism, but then at the same time, we don't want to be hamstrung by pessimism, do we?
No.
So, as always, Oliver likes to walk that line like Johnny Cash.
I walk the line.
Sometimes
I fall off, but I try to get back on it.
He's a bit wobbly sometimes after a couple of beers, but he always tries to stay on that line.
Okay.
So I think that pretty much wraps it up. The long and short of it is, CRISPR, it ain't over yet. Watch this space. And we'll be back next time. And speaking of which, we'd like to thank our patrons, the Lifespan Heroes, for continuing to support us and helping us produce shows like this and many other things. And if you're interested in becoming a Lifespan Hero or learning a little bit more about that, if you visit lifespan.io/hero, and you can learn a bit more about becoming a patron and supporting us. And that's it. And thanks for everybody who's joined us. Thanks to Oliver again for showing us the way through the labyrinth of science. And we'll be back next week. Next week. Good grief. We're not there yet. Don't worry, folks. We're not doing a week.
Yeah. And let's figure out the time, because I know a lot of us are taking vacations in August, so we'll have to kind of maybe think about doing it first week of September. No, too late. Last week of August.
You know the rule. We can't possibly do a journal club for 1 month in the following month. Okay, we did it once, but that was only because of a series of unfortunate events.
All right. Well,
we'll see anyway.
Well, in about a month, we'll be on air live again.
Yeah, we'll see you. We will see you. And if anybody does have a suggestion for papers or anything that you'd like to hear us talk about, get in touch.
Okay.
Right.
All right. Thank you.
Thank you very much, everybody. And we will see you next time. Bye.
Bye.